to think about potential impacts and get this site permitted. Oh no, now I can hear myself coming through this.
Oops.
White cells are unlikely interactions. Green ones might be very unlikely or low impact, low consequence. And then yellow or red ones are those high consequence interactions. Yellow or red squares, we can choose an assessment approach, so how might we assess those interactions and decide on metrics for determining the impact, what habitat criteria or what indicator species. So on the continental shelf off Oregon, we don't actually know a lot about what might be important habitat features or indicator species. Because while we have a lot of great mapping of our shelf, because of the work of many researchers here at OSU who've done substantial mapping and geotechnical surveys of the shelf and the slope over the years, we haven't done a lot of ecological. There's not a body of literature about the ecology of the sedimentary shelf of the Pacific Northwest. Okay. So what would be the criteria and indicators that we might use to assess these potential impacts? Okay. Can I hear myself on this video? Okay. All right. Bring us here. What are the criteria and indicators for impact? Or, well, how would we determine what is an ecologically significant change in habitat or community from my carry out on the shelf? As I mentioned, we've got these great maps of the seafloor on our shelf. And traditionally they have been called habitat maps, but they in fact are not habitat maps. They're geologic maps. Because the boundary between classes have not been defined based on any known. So the first years of my time here at OSU were really focused on developing these habitat maps for these four communities. PGs, as my faculty probably call them, the investigations. We use a box score where we can actually get samples of the organisms and their habitat, analyze the sediment, use UCAS to get water quality measurements, identify the impoundment in the lab. And then we can take a couple different approaches for determining what habitats, how habitats might be defined for these kinds of organisms. One approach is looking at individual species. So doing some habitat suitability modeling. So here, Grassy, Sandy, and Andrea took a Houston regression tree or approach, Bayesian approach, making habitat models for individual species. And then we did a number of different projects using a technique where we define assemblages of organisms. So what organisms are frequently found together, certain suite of animals, and then figuring out which habitat criteria differ amongst those organisms. And so at first we did this far left, just in the Pacific Northwest. just from 50 to 110 meters water depth, really targeting those areas that at the time were proposed for wave energy development. Then Walt Nelson from EPA and I took advantage of the EPA data set from 2003 that meant the entirety of the US West Coast and looked at a slightly broader depth range throughout the California current. And then when there was an offshore wind farm proposed off Coos Bay way back in 2014 for about 500 meters of water depth, we took additional samples out to 500 meters throughout Oregon and developed these habitat classifications ranging from 20 meters. And there we ended up coming up with 12 different habitat types in an area that maps that were called habitat, but really were just geologic types. So this has really formed the basis for what we would call a early 21st century baseline of what the habitat features and organism distributions were. And we're really already seeing changes to the distribution of organisms in these habitats due to global change, which we'll get to later in in these three studies, we mostly were looking at environmental correlates that correlated with, in some cases, we've actually been able to get at the mechanism. So one of the distinct breaks that we see in invertebrate distributions is about 45 to 50 meters water depth. It's pretty consistent, and doing a little bit more investigation, we determined that that was likely driven by the winter storm wave base. So the depth at which the energy from winter storms can be felt on the bottom, and if you're deeper than that, you're an organism deeper than that, you're not going to be affected by the mixing from winter storms. For bivalves, and so here, maybe this one of my favorite little clams, Axonopsida, could become an indicator species for changing wave conditions. It will not go shallower than that winter storm wave is. Does not want to feel the energy of the waves in its habitat on the bottom. So yeah, we're trying to figure out how we might assess and determine impact from offshore renewable or other projects. So to detect habitat changes relative to macrofauna, monitoring, you must be able to detect trace amounts of silt or clay or fine materials. So one of the things that we found across those studies is that as little as 8 supports a different assemblage of organisms, 4% sand and 6% organisms are completely intolerant. And so if we want to see if we're having that kind of change, we need some sort of assessment method that's able to detect those fine scale changes. And so we might not be able to use remote sensing techniques. Acoustic backscatter is not going to distinguish the difference between 2% silt and 0% silt. What does this mean for benthic communities? So if you've already got a silty area, changes in the silt may not make a big difference. But in sandy area of the macrofauna communities, and then as I discussed for our poster child Axonopsida, changing wave base due to either changing storm conditions associated with climate change or wave energy extraction could result in a change to communities. So now we've got some criteria, we've got some indicators. We also wanted to look at megafauna, and so here is where we use visual methods. We've got the submersible delta, we've got an ROV, we've got USGS and bobsled. We've used all of these techniques done by my very first grad student here, Tim Lee, and my very productive postdoc, Leda Canary. Find these habitats based on fish and invertebrate assemblages on the shelf across Washington, Oregon. In addition to defining habitat characteristics, we also took those data and did some more habitat suitability modeling to look at the distribution of sea slot formations prior to the event. And so now we have a baseline that in the future, as we see these populations recover, we can look to see if the distributions and abundances and the habitat for these stars has returned back to what we might have expected to have been before that racing event, or if we've got a different trajectory coming on. Again, once we had the area of the proposed wind project in 2014, we expanded our sampling down the slope and wrote another paper looking at fish and invertebrate assemblages on the slope. And while we were out there, we discovered this really unique cobble pockmark habitat. So, and what we previously thought was broad sandy seafloor, there were little teeny tiny methane seeds, just green bubbles coming out with bubbles around them. And they were hosting these crinoids or thunderstars, which they themselves ended up being structure forming invertebrates and facilitating rockfish recruitment in this area that we thought previously had been broad sandy habitat. So one of these findings, this body of work on now habitats on this floor mean. So it does seem like these substratum types can be mapped or monitored using remote sensing. That's what we used. With remote sensing, we're able to visualize thin veneers of sediment on smaller on bedrock, but that thin layer of sediment did not result in different communities there. So for benthic communities, small changes in sediment deposition on bedrock habitats might not result in ecologically significant changes in epibenthic communities, except in these unique cobble park mark habitats. They're so small, if we were to sediment in those cobble habitats, we lose the structure. Okay, so now that we've mapped and assessed and figured out habitats on the Pacific Northwest Shelf, and so what can we do with that information? We've got lots of questions about the potential effects of radioactive installations, but nothing yet to study still. So what can we use as proxies? What kinds of human uses or other activities can we use to maybe think about potential effects? And also, are there opportunities to study other human now? And so over the years, there's been many of these. And so if you're watching this online and I don't talk about your project, something had to make the cut. So apologies to those of you whose project didn't make it into this presentation. So I'm going to highlight two of our offshore disposal projects that we did. And so pictured here is grad student Stephanie Fields, who now works at ODF&W. And her project was looking at effects of dredged material disposal. So dredging it out of the Columbia River and depositing it with renewable energy with some changes to the sediment deposition. And so we use this as proxy and also just think about the effects of this. And so we had two research projects affiliated with this. One was using, again, a towed camera sled before and after and in impact and reference areas of the dredged material disposal to see if we get different epibenthic communities in the disposal site versus reference areas. And the quick answer is not really. These communities are pretty resilient. One of the awesome things about the Pacific Ocean is it's really energetic and really dynamic, right? And so, as it turns out, if you drop a bunch of sediment at the surface, 60 meters later, it's pretty wet ocean, and so we don't see a ton of mounding of sediment. We're not smothering the mountains. The other element of this project that was absolutely the most fun thing is we put down these baited video cameras. So video cameras, and they've got a bait jar at the bottom. And crabs love bait. They really love bait. And so the crabs all came around our little video lander. And what you can see pictured here is we would set up our little pods. We put flags and if we string so that the dredge would know exactly where our video cameras are. And we just open the doors. Down comes the sediment right on top of our video pods. So what comes first before the sediment comes is a giant pressure wave. And so turns out the pressure wave blows the crabs away and they don't even encounter them. But they're gone, they're long gone. The sediment comes down, it blacks out the camera, the sediment settles, and the crabs come back. Yes, they love it. So this was an extremely effective project. Crab fishermen were really worried that their crabs were getting smothered with this disposed material, and then that maybe the crabs, wouldn't even come back to that area because it was so traumatic for them. And it turns out if you just put bait down, the crabs would come. And if anyone wants to follow up, all those videos are posted on YouTube. So if you want to see that, reach out to me. I'll give you the link to it. The other offshore disposal project we did was looking at the GP outfall pipe. So the Georgia Pacific paper plant that's in Toledo actually has an outfall pipe that goes like all the way out to the ocean from Toledo to Newport and offshore. And some earlier studies had not found any differences in the benthic communities near the outfall pipe versus away. But Newport citizens were still quite concerned about this outfall pipe. And so we got money from the city to collect lots of species of interest, crabs or a seafood species of interest, English sole, as well as non-seafood species. And we collected them and sent their tissues to Vices Laboratories, which is extremely high precision. They looked at 90 different parameters, so metals, DDT derivatives, PCBs, PPDs, and to see if we could see any chemical effects or bioloading from the GP outfall pipe. And so here's just one of 90 parameters, cadmium and Dungeness crab. And everything was well below this line is the FDA level of cadmium. Here is a non-conservative total recommended value, and then they were above this ultra conservative. But what was interesting is the mixing zone is this middle bar. Never amongst like what was almost always the highest, not in this cadmium case, was our South Beach reference site, which turns out is really close to where they dispose of the Uquinna Bay dredge. So maybe no physical effects from the sediment deposition, but raises some questions about what might be in the Uquinna Bay sediment that then organisms that are living in that area might be making after. Opportunity for future questions. More recently, we've looked at some other proxies in terms of noise, so both acoustic noise and EMF noise. And so featured here is one my co-author Taylor is in the room. And so was mapping the Cascadia subduction with a 36 air gun array. So it was pulsing these air guns, bouncing them off the seafloor to map the subduction zone. And it was going past the Red Fish Rocks Marine Reserve 3 times. So we had previous fish tag data, rockfish that had been tagged in the Marine Reserve. And so we thought, oh, this is a great opportunity to look at crab and fish response to noise. This air gun survey, this seismic survey, and also maybe learn some lessons about energy project. So we had maybe the most dense array of Benko receivers ever deployed. We had 45 Benko receivers in the Red Fish Rocks Marine Reserve. We tagged 15 each of lingcod, black rockfish, China rockfish, and Dungeness crabs. And the lingcod left. So see you later, $300 times 15 fish. Rockfish stuck around, and we got some data from the crabs too. Both the rockfish and the China rockfish did increase their acceleration on the days that only marginal for the black rockfish, but for China rockfish who don't move, those acceleration changes were pretty significant relative to how much they move. The other thing we hypothesized that happened with the black rockfish was that they would collapse to the bottom, has been documented in other papers. Stress on rockfish goes to the bottom. We looked at our data, and our rockfish actually missed up in the water column on the days of the survey vessel went by. We thought, well, gosh, that's weird and it seems counterproductive. And then when we did some 2D exploration of their movements, it turns out that while on the first panel, the rockfish are normally just distributed all throughout the Marine Reserve, on the day that to the middle of the reserve, which is where the reef is, and so they went up in the water column, they were going up into the rocks. And then after the survey passed, they spread back out through the Marine Reserve. And so for both the rockfish species, we did detect some acute effects, but they didn't last. They didn't normal. The other kind of noise that we worry about when we think about offshore renewable energy is noise from electromagnetic fields. So we're going to have cables from the devices to the seafloor, from the project site back to land. And so there are many, many questions when it comes to animal responses to EMF. And so we began this by doing some experiments in what we call the fish barn at Hatfield. It's space that belongs to the Alaska Fisheries Science Center, so we really appreciate them. And we started with some species that we were pretty sure were electromagnetosensitive, so skates, and then ones that maybe were Dungeness crabs and red rock crabs. There have been some conflicting information in the literature, but they're species that people really, really care about. So we thought we'd start with those. What you see pictured here is inside the fish barn, the tank inside this huge cage of cables. And our postdoc, Kyle, figured out how to turn on the right cables and in what orientation to geomagnetic the magnetic field, the strength and the inclination. We could make it magnetically seem like Alaska or Bob. So we thought this might motivate the animals to move because they were like, I don't belong in Alaska. I don't belong in Baja. I belong in Oregon. So we wanted to see if first they would respond to that. Then we wanted to see if they would respond to the cable being on under the tank. So we had the cable under the tank, we put from AC through it, DC through it. And then to really get at what the concerns are, we wanted to see if the cable being on or off changed their response to the geomagnetic displacement using the magnetic fields. And we turn on this cable that could be really confusing to them. Will that disrupt their migration? So what did we find? The crabs did seem to respond to the GMF stimuli. So maybe some evidence that they could at least detect this stimulus. We still can't say in a 6-foot tank whether they navigate using this stimulus, but they certainly seem to respond to that. When we turn the cable on AC or DC, the Dungeness crabs kind of slowed down when it was on direct, whereas the red rock crabs definitely slowed their excursions around the tank under both conditions. And so we don't know if this is, they're just slowing down and exploring, or this is a stress response yet. We've got to do a little bit more work. But in either case, they didn't avoid the area of the tank where the cable was. So it wasn't necessarily aversive to them. And in fact, there's some evidence that the red rock crab liked the cable, kind of spent more time sitting on the cable part of the tank. Similarly, we saw the species, the long nose and the big skate, detected and responded to the GMF stimuli, the Garrett coils. Both of them slowed down when we had the cable running DC, and the big skates moved around faster and more when we turned it on. So again, this is a sampling or habitat scenario or stress. We're not sure, but again, either skate avoided the area of the tank. Now, as I mentioned, this is a relatively small tank. And so what's happening now, which is super exciting, is Kyle has moved over to the Oregon Coast Aquarium, where we have a humongous tank, and animals who have lived their whole lives in captivity, and so their behaviors will make it more normal in a tank. And so far, we've been working with dogfish, sharks, and bat rays, the cable under that big tank. So exciting results to come, I'm sure. All right. So now I want to transition to where I think thematically the lab is going in the future, looking at effects of ocean change on ecosystem engineers. In terms of instead of just whatever species I happen to have opportunity to study, I'm really trying to focus in. And this was precipitated by two events. One, in 2019, so remember 22 slides ago when I showed you the Pathway project, in 2019, we finally got the license for that. So it was time to go sample that site intensively so we could really understand the variability within the site, so we could maybe tease out effects of a device deployment. The other thing was in 2020, my RU from 2017, whose name is probably familiar to most of you in this room, Angelina, was wrapping up and doing a PhD with you on intertidal health. And 2020 was when I finally transitioned to tenure track and was allowed to take PhD students and had support for PhD students. And if you've known me for a long time, I actually did my master's and PhD in health. And so, yes, come to OSU, be my very first PhD student. Let's do some health work again. And so this really got me back in the intertidal and back to the cups. But why did this first one turn into a project on ecosystem engineers? Well, like I said, it was time to intensively sample the package set. And so what might be hard for you to see, but those are like about 1,000 red dots of where over the years in preparation for the package project actually existed. And they're pretty spread out because we wanted to see, well, is the Pathway site like the rest of the shelf? Now that we've got our project, let's get in there and hit all these white dots and really figure out what the site looks like. And then I told you that to sample these like small portions of silt, we need to actually get the set. But we thought, yeah, why don't we try something new? There might be another tool out there that we could use. And so that tool is the SPY camera. And this SPY camera is, SPY stands for sediment profile imaging, and it has this balance weight which will trigger the plan view camera before it lands on the seafloor, and it lands on the seafloor, and this prism with the camera inside slices in so that you can see there's a very small inside of the spine, which the sediment itself, like down in the sediment. So we thought, well, maybe we could use this to get at these small portions of silt. Not entirely remote, we are penetrating the sediment, but not collecting anything. And then we were going to ground truth with box score samples and compare the images to the box scores. And so we dropped this thing 50 times, and then we got off and we downloaded the data, and it's on the seafloor with the plan view camera. Giant holes, like way too big for anything I had collected before. We were like, oh man, what is out there? Who did this to our site? Like, we haven't even done anything there, and our site looks totally different from the last time we were out here. So we did go back and we ground truth in the box score, and then we got tons of these ghost shrimp. The ghost shrimp, they're well known from the estuary. We've been studying them in the estuary for decades. Every time a school group comes out, we take them out with the shrimp slurpers and slurp up some ghost shrimp. So we know a lot about them in the estuary, but they're not supposed to be on a shelf 7 miles offshore, 70 meters of water. Like, what are they doing out there? So there were a whole lot of questions. So we went out and we sampled up to as many as 26 shrimp in a 0.1 meter squared box. That's 260 shrimp per meter squared. There's a lot of animals. And they all make these burrows. And so we reported this and we made some estimates. So we look here, the number per meter squared on average was about 40 when we went out in July, and when we went back September, October, we got over 50, which is rivaling the densities in Willow Bay back in 2006, and the bays have all been declining since then. And here we are finding tons of them out on the shelf. Because of their burrows, this is a really back in the envelope calculation, we thought they might have increased the surface area by about 100%, 70 to 130% because of the sizes of the animals and their burrows. And so in addition to these animals being large, they're completely changing the structure of the seafloor. They're actively maintaining these burrows, moving a lot of water through them. And so I reached out to Claire Reimers, who's here today as well, and said, Claire, this is a big deal. And you said, right. So we wrote this proposal to NSF, environmental consequences of expanded recruitment of an ecosystem engineer on a hypoxia-influenced continental shelf. We got all the right words in there. And it was like, yeah, we agree. So we've been working on that together for the last couple of years and have a lot of questions about this. So one of the first questions was like, who are these shrimp even? We got 2 species in the estuary. Pictured up top is neotropic gigas. And one of the things that distinguish eye stalks, the gigas have concave edges, whereas the Kelpinians have convex. So you kind of look at them quickly and see which ones they are. So here's our offshore ones, and the edges of their eye stalks are pretty concave. Well, gosh, I mean, they mostly look like gigas, but they're not exactly. They're just so weird about these shrimp. And one of the things that Sarah Hardesty, who is behind this, notice, one is that if you look at the pigmented area of their eyes, big black pigmented areas like the Californiensis. So Sarah and then one of my RUs measured 29 different traits, so measured 29 parts of these shrimp under the microscope and did a linear discriminant analysis cluster. And they are distinct from the estuary. So we're right, they look like gigas, but not exactly. So then we did some genetic analysis, and genetically, they are gigas. You can see here, this is the 18S gene. So the red ones are all the Californiensis, green ones are the gigas from the estuary, and the blue ones are the offshore shrimp. No difference with this marker or the 2 other ones we tried. So for now, they're gigas. Just little. So, how do their burrowing behaviors compare? Like I said, we know a lot about them in the estuary, we've been studying them for decades. Can we take those lessons from the estuary shrimp and apply them to offshore? Can we estimate the same volume of water moved through? Can we estimate the same increase of the surface area of moved a lot of buckets of mud? Many whaleboat trips to the estuary to bring them back to the experimental seawater facility at Hatfield to fill up tanks. Many coolers of mud carried from the boat back to Hatfield, and then poured resin down the holes and entombed the shrimp for perpetuity and made these resin casts. And so pictured here on the left is a resin cast from an estuary shrimp, and on the right, an offshore shrimp. And you can see I made the ruler line up. The estuary shrimp burrow is much longer. And in fact, like my first idea when I saw this picture, it just hit the bottom and then like went to the side. But Matt also set up ant farm tanks so he could video their behavior and estimate the area of oxygenation. And you can see again on the left is the estuary, and on the right is the offshore shrimp, and it had plenty of more distance. It could have gone down in that ant farm, and it just didn't. It made that J shape at the bottom. And so you probably can't use estimates of the morphology of the estuary burrows to apply to offshore. Might be moving through. You can see the area of oxygenation in this black sediment around where the shrimp are. Also, what's interesting is if we look at where within the burrow the shrimp are hanging out, still the ones that are offshore shrimp, the estuary are the red ones. Most of the time, the estuary shrimp, even in this completely artificial environment where there is no tide, they're in a ant farm, the estuary shrimp are going to stay down in the middle of the bottom of their barrow, and they're not coming up to the surface. Whereas our offshore shrimp, who are used to being in 70 meters of water and no low tide, no birds to snatch them, they'll just hang out in the top or even in different burrow structures and burrow meters between these two animals. Are they elsewhere offshore? Did this expanded recruitment only happen in Newport, or is this a coastwide thing? So again, Sarah Percy, she did a lot of species distribution modeling, coming back to that technique. She actually did quite a bit. She took 3 different input data sets and did 2 types of species distribution modeling. One BRT, which is a presence absence method, and one MaxEnt, which is a presence only, to try to see where these shrimp might be. We took these 5 different models and overlaid them to find where they might agree there could potentially be hot spots for shrimp. We found a number of different sites. And this is the predicted suitability on the bottom. As a reminder, our pathway site is where we first found them. But there were some other sites that seemed like potential for shrimp. And then we got on the Pacific Storm, and we went all the way up the coast. And these are our data of where we actually got shrimp. So the 2 sites in Oregon, Nehalem and Neskowin, that had high predicted suitability, we found shrimp. And then the 2 sites in Washington that had high predicted suitability, we didn't find shrimp. But we did find some at a site off Willapa that had lower predicted suitability. So now we've got 4 sites along the coast where we know these shrimp are. So now it's time to get on a big ship with Claire and her team and go figure out what are their chemical impacts. What are the plastic changes that we might see. So Claire's got her eddy covariance lander and her slow core to collect chlorus for incubation. In that incredibly small diameter core, we did stab a shrimp, which was really exciting. We got one in the slow core. We also took along my video lander because we wanted to make sure that we knew if it was a shrimpy area or not, even if we didn't actually grab a shrimp. So we took the video lander so we could check for the holes, so in case we didn't grab any. So what Claire's team found is that the core incubations showed the highest oxygen and nitrate uptake rates at sites where the shrimp burrows were abundant. So our Nehalem site, where we had the most shrimp burrows, also had some of the highest uptake rates. And then the flux from the EC lander was highest at the places where we had the most shrimp in the box score. So definitely these animals are having a pretty substantial impact on the movement of water and thus oxygen and other nutrients across that sediment environment. Finally, back to my squidgies, as my friend calls them. What are the effects, the community impacts? pretty substantial. So if we look at the worms, the crustaceans, and the bivalves, you can see in red are the picture or the stations where shrimp are present. And in black, the stations where the shrimp are absent. We're seeing quite different communities in those three subsets. The gastropods, not so much. Snails, nah, they don't much care. The shrimp are there, we're not. One of the ones that we've had the opportunity to look into so far, which might be Axonopsis auricata was my favorite little clam because it was a good indicator. But this Pridia lincivita had been a pretty minor player in our observations up to this point, and even at the stations where we didn't have any of the native trichaea, we get maybe about one per box score. But where the shrimp were present, there were outstanding numbers of these clams. 20, an average of 20 clams per box score. And in some cases, we had like over 40. These pretty LLGs. So just starting to look into this, but doing some literature on this little clam. It's been found to have a positive association with the aerobic halo of a tube-forming polypete. So that seems pretty consistent with our findings about the burrows of Neotrichaea. And then its North Atlantic congener is positively correlated with the oxidized layers around the burrows of Rostock. So seeing some clear analogies there. So we'll continue to look into this, which worms might be affected, what are the mechanisms, why aren't gastropods affected? And then, of course, we have future research questions. So why did they settle on the mid-shelf? We did a cohort analysis when we first found them and think that they probably settled around 2014 or 2015, which were kind of impactful years around here. We had the heat wave with the blob. So hypotheses, maybe they had increased development rates due to the warmer temperatures, and thus were ready to settle out of the plankton before they made it back to the estuary. They normally come out of the estuary and go back in to settle, or maybe some oceanographic fronts kept them from returning to shore. So opportunities for some lab experiments on larvae, some hind casting. Why aren't they in between? So this is an estuarine species. I found it at 70 meters. I've been out there stabbing the bottom ever since, looking to figure out their distribution, and they are not shallower than 55 meters. Look at all those nodes. And so this makes me think of our friend the clam, who didn't want to be affected by the winter storm wave base. And this paper is not mine, but I could not resist. Props to Ranky and Appendini for putting 2 puns in one very short title. Okay. But I think the wave base has something to do with this threshold of the shrimp distribution. Promised talk about macrophytes in the intertidal, but I only have a couple minutes left. But we are looking at surf grass phytosphenix. How will future ocean conditions impact the relative dominance of these 2 occurring foundation species? This project was motivated by this long-term data set by Marine, looking at how these 2 species aids. So we're investigating research questions about how do the understory compare between these 2 macrophytes? How will their performance be impacted by changing water conditions and nutrient conditions that are associated with climate change? And how will these communities change if we get a flip-flop of the... My last ecosystem engineer story that I left as a teaser is one that I love because it's something I've been thinking about a long time and really has engaged a lot of students over the years. So sand, their distributions are also dependent on the environment. What little is known is that they live in like less than 20 meters of water, and they go deeper in the winter because the waves can go deeper, but in the summer, they have to move shallower because of suspension feeders. They need that energy to keep food delivery to them to pull out of the water. Yeah. So I always thought maybe someday this would be a great indicator for an ecologically significant amount of wave energy extracted. You can change their distributions so they extracted so extracted any energy. So I haven't gotten to use them for that. But I've been keeping an eye on them. And one of the ways I've been seeing bruises. So Oregon State University, every biennium applies to the Ocean Going Research Busing Program to get days of student take high school and community college students. So since 2018, I've engaged about 288 high school and community college teachers in marine research and also the Pathway story. And I just always take them out to the Pathway site, tell them about the project, and then we're taking care of things, right? We're checking on things out there. Where high school and community college students have taken those samples. I use those samples in my summer class, my 5 through 53 class, and those 5 through 53 students get to process those samples. Either the box score samples, image J to count and measure the sand dollars. So here's the student's lab report from 5 through 53 using the sand dollar data. And so it's just been an awesome opportunity to engage through grant writing. But I think it's time to write a grant. So while we see pretty low abundances of the depths that we're working on, skip some time, 19 and 20. We had one big sand dollar year in 2014. They're not super abundant at the depths. Really thought they stayed pretty shallow. Well, these are the data of late. The sand dollars are blowing up and they're also moving deeper. And I made this plot, and then last week we went out and in one box score, we got 1,208 sand. I'm calling them dimes because they're so small. And I decided 500 the first panel, 500 the second panel. That's how many sand dollars. So if your seafloor used to be sand, and your seafloor is this now, that's really different. If you're a burrowing organism, or if you were an English sole and you wanted to like nibble little worms or the sentence of plants on the surface of the sediment, you're out of luck. There's no surface of the sediment. There's nobody else in there. So this is really another dramatic change. So some future work is the OI benthic lander is out there. It's at about 45 meters, and I'm standing the images, and you can actually see there's sand dollars in the images, and then they're gone. And then they're in the images, and they're gone. So I've been working with Art Trombonus at University of Delaware to train we can go through those like thousands of images from the OI solution to track the changing distributions. So what about offshore renewables? We do have one wave energy device in Oregon, not in the water, in the shipyard. And floating offshore wind does exist, but not in Oregon. This is a picture of me in Scotland back up. It's right in the back. The Pacific Marine Energy Center had gone from nothing being in the water to we now have cables in the water. So the HMS Innovator laid the cables down off this giant thing that looks like a roller coaster. And then we've been using the underwater ditch witch. That's not its real name, but that's what we're calling it so that community members understand it's an ROV on track that goes back on top to bury the cable. And I mentioned I was out last week collecting 1,200 sand dollars. And here is me actually box scoring, trying to determine the effects of the cable. And then my new student, who maybe some of you have met, MJ, will be looking at assessing the growth on devices once we get them out there. I finally can do the job there too. That's the environmental effects of offshore renewable energy. And so in addition to vapor and wind, got some seed funds to do to submit as sort of excellence proposal. And so hopefully after we submit that on November 7th, which is really soon, we're running to be in addition to a Marine Energy Center of Excellence here at OSU, also an office. I will skip these further. Thank you.
So I may have missed this. So back to the shrink, the ghost. So they're in the whatever it is, the estuary, that's what I said, the estuary. And they've been there for years, but they're declining. And do we know that they weren't out deep, or no one ever went to look for them?
Well, so you can see box scores,
Yeah.
they weren't. And then in 2019, I did.
So you were, yeah, so you were looking or whatever, and so that's good, and so. So you're not telling us, but what's your thought? What's the deal? Is that global climate change? Is that, there's some stochastic thing?
Yeah.
What's your hunch?
So like I said, when we did the cohort analysis, it looks like they settled the blob, so we like to blame everything on the blob, so let's just give it this one too.
Yeah.
And so I've got two hypotheses which may be complementary, is that one, so the shrimp in the estuary, they spawn, there's larvae, the larvae go out to sea, and then the larvae are supposed to come back to the estuary itself. And so it seems like, so either maybe the warm water may settle, the ocean conditions were turned, this lens of warm water, the cold water on the bottom, it couldn't get past, and so they trapped offshore.
And then to just keep, so they'll ongoing, right? They're not living for 10 years, like so whatever happened, it's self-sustaining.
Right, so
And there's not a blob out there.
So the question is now, is the offshore population exceeding itself? So that is another question. So we, when we did the morphology and the genetics, Sarah measured a bunch of shrimp that were the ones that we collected in 2019. And then ones that shell measured were ones that we collected, and they didn't cluster with estuary, so that's like, but again, more opportunity for future work, yeah.
So, and I might have missed this, but when we were like, okay, they could be here, they could be here, they could be here, they could be here, and they're only in these few spots. We went from that, what brought you into looking at chemicals, and did that show you why they were where they were, or?
Good question. In this habitat, we have very low oxygen on the seafloor. And so we wanted to see if these shrimp were at the one site, but since we found them other places, we did that at all the sites. The interesting thing about when we finally did go on our trip and find them at other places, well, the model had lots of, the places that we found them was always south of an estuary. Like, even here in Newport, we keep going back to the NH line because there's lots of, they're not north of Uquinna Bay. They are only south of Uquinna Bay. And then we found that with Tillamook, and we found that with Willapa. So some getting trapped, not making it back, seems consistent. Another project that I didn't have time to include, so thank you for the microbial communities, both in the estuary where we find them and offshore, and in the estuary where we don't find them, and offshore where we don't find them. And of course, like the microbial communities in an estuary are very different than offshore sediment, except in the shrimp positive stations in both of those, we actually have overlapping microbial communities. So I don't know if microbes attracted the shrimp or the shrimp are hosting the microbes, but there are microbial community similarities that are in shrimp positive both in the estuary and offshore, which is super interesting.
So just out of curiosity, on shelf areas, has there been increased amount of sightings of great, kind of focusing more on ghost shrimp as an eating option and like that going into estuaries and kind of risking it and risking stranding? So, and it seems like this would be a pretty nice spot where plenty of depth so that they wouldn't strand and pretty high.
Not yet. This is one of those, so good thinking, great work. So they do eat ghost shrimp and the ghost shrimp. Now remember, 5 meters, and most of our gray whales, when they're a little bit further offshore, they don't eat, they're going down there and mate, they're just looking at it. When they come back, they're going shallower and they're feeding. And so they shouldn't encounter them unless they just go a little bit further offshore. So no one has observed it yet, but Lee, Torres, and I, we talk a lot, and hopefully it's okay. And Charles Knight is doing DNA in the feces of the whale. So when they scoop up the whale poop, he's sequencing it and looking for string. So he's reacting. . 。
Okay, let's, oops, sorry.
Shrimp. Sorry, I'm captivated by these shrimp. So you had a chart that had them, that had circled, have the genetic map of them, it's saying as something that they're not the same in this area, is that because their genes like aren't expressing the same, or is it because like would those two species be able to, are they the same, like could they, if they bred, would they be viable offspring, like?
So the Neotrophagous and the offshore shrimp, genetically, from the 3 markers we've used, indistinguishable. Absolutely, I think they could reproduce. You keep sequencing. And we might find some marker that from the difference. They don't look exactly the same. So whatever morphologically is different about them must be something that is within the range of plasticity, especially, I mean, I think about the pigmented eye spot, you know, you can add more pigment without changing your genetic code. But yeah, and then what will be interesting is if in fact this population is self-seeding and not interacting with estuaries.